Good morning, good afternoon, depending on where you are. Thank you for joining us for this webinar sponsored by ACEA Biosciences, which is now part of Agilent Technologies. My name is Yama Abbasi. I'm the Vice President of Business Development and will be your host for today's webinar. Before I introduce our speaker for today, allow me to share a bit about Agilent Technologies and the products we offer for cell analysis. Agilent is a trusted leader in life sciences, clinical research, diagnostics, and applied chemical markets, providing laboratories with instruments, services, consumables, applications, and expertise to enable our customers to gain the insights they seek. ACEA is one of the newest additions to Agilent's cell analysis division, which includes Seahorse, xCELL, and more recently, Biotech. Agilent's rapidly growing cell analysis portfolio enables deeper, more reliable insight across a variety of cell analysis applications, where investigators and drug developers seek to understand complex cellular environments and interactions, including cell fate, fitness, and function. Our comprehensive cell analysis tools are critically important in various investigation fields such as cancer, inflammation, immunology, immune oncology, and as you will find out today, infectious diseases. I am absolutely delighted to introduce today's speaker, Dr. Brandon LaMarche, who is Head of Applications Development at ACEA Biosciences. Before I hand over the mic to Brandon, I would like to remind you that this webinar is interactive and that there will be a question and answer period immediately following the talk. You will notice on your screen that you can, and in fact should, type in your questions during the presentation in the question and answer field, as this helps ensure that we get as many of your questions as possible. With that, thank you. And Dr. LaMarche, please take it away.
Thanks, Yama. So I'm going to be talking about real-time high-throughput viral cytopathic assays that can be used for vaccine development, neutralizing antibody screening, and antiviral drug discovery. So I'm going to start off today by discussing the principle of cellular impedance, which is the technique that the xCELLigence instruments use. I'm going to first look at this at the level of first principles. And the reason for this is that if you understand at its core how the assay is working, it's very easy to then think about how you can incorporate it into your own workflow, including applications above and beyond those which I'm going to describe today. I'll then look very quickly at an example of why impedance is so useful for virology assays. Collectively, this will serve as a foundation then for looking at some specific applications from the literature, including a few different examples of neutralizing antibodies, virus titer determination, antiviral drug discovery, and then looking at a comparison of different levels of viral fitness. I'll finally close out by looking at our newest instrument in the xCELLigence line of instruments, which is called eSight. And this takes the real-time impedance monitoring and combines it with live cell imaging. So I'm showing here an example of one of the xCELLigence instruments. The line of instruments primarily vary from one another in terms of their throughput. The one shown here has 6 different cradles. Each of these cradles accommodates a 96-well plate. And these plates look very similar to a traditional microtiter plate. The difference is that they are electronic in nature. So if you look at the underside of this plate, you'll see that all 96 wells have a gold biosensor array that is integrated into the bottom of the well. These plates come with either glass or plastic bottoms. I'll talk more in detail here in the next slide about how these biosensors are working. But for the moment, what I'd like to say is just that you would load cells and virus and drug into these wells as you would in any other assay. The plate gets put inside the xCELLigence instrument, which which is housed inside a regular tissue culture incubator. So you're going to control temperature, humidity, and atmospheric composition as you would in any other experiment. The xCELLigence instrument then interfaces with a control unit that sits outside the incubator. This computer runs the xCELLigence software. So this is where you program the details of data acquisition. It's also where you can watch in real time your data being collected. It also includes a suite of analysis tools that can be used for producing quantitative information from the time data. So what I'd like to do now is take a zoomed in view of a single well from one of these electronic plates. Showing here a side view. The well already contains media in it. As I said, there are gold biosensors. Sometimes we refer to these as electrodes. They're integrated into the bottom of the well. When you put this plate into the xCELLigence instrument, it provides a very weak potential, and this causes current to flow from one biosensor to the next. The current is literally flowing through bulk solution. And this measurement in the absence of cells serves as the background reading. And what it's teaching the instrument is when it applies a constant voltage, exactly how much current can flow through the system. So this will be dependent on the composition of your growth medium. The higher the ionic strength of that medium, the more current will flow through this. So having made that background measurement, you can now add your cells, and of course they'll settle out and attach to the bottom. This is physically a much different situation. Cells are inherently insulators, meaning that they resist the flow of current. And so another word for that would be impedance. They impede the flow of a current from one electrode to the next. This is basically what xCELLigence is doing in a cell-based assay. And some of the key features of this system are that you use standard tissue culture media. No special labels are required. And adhesion and proliferation on these gold biosensors is normal. We've shown that now over years and hundreds of different cell types. Also important is that the cells are not affected by the electric field that they're exposed to. It's very, very weak, and it's also not applied constantly. So you can acquire data once every 5 minutes, once every 5 hours, once every 5 days. It's just whatever it is that you're interested in doing. But the cells are not exposed to the electric field constantly. You're going to see throughout the course of this talk the benefit of real-time monitoring. Another important feature of the impedance approach is that it's compatible with using orthogonal assays. And so what I mean by this is that the process of measuring impedance does not destroy your sample. If you so desire, at the end of the assay, you can collect cells from the electronic plate and then use them for example, Western blotting or RNA sequencing or whatever it may be. The last thing I'd like to say about this is that the signal that's being reported when you're measuring impedance is a term we call cell index. And so this is simply the impedance at the time point of interest. So let's call this 10 hours, then subtracting out the background impedance signal at t equals 0, so prior to adding the cells. The situation I'm showing you here is a bit of a simplification. There are indeed just 2 electrodes or 2 biosensors in the bottom of the plate. However, they're arranged as an interdigitated array, as I'm showing here. So one of these is in black and the other is in blue. And the benefit of this configuration is that it allows for us to cover about 75% of the well bottom with the biosensor array. And the benefit of that is that it allows for you to measure thousands of cells simultaneously. And this gives you very high sensitivity. The plate type I'm showing here, in this photograph, you can clearly see the gold biosensors. The biosensor free region in the middle of the well facilitates microscopy. So I'm showing here a few different examples of the way cells have been imaged these plates. So that's the xCELLigence instrument, and that's the plate. That's what we're monitoring. The most important question is, what's it good for? What kind of questions can you answer using this approach? One of the things that you can monitor is changes in cell number over time. So if you look at a situation where you have a small number of cells and they begin proliferating, this is a much different situation. So More cells covering more surface area, it's going to have a higher impedance signal. And it's important to recognize that the process is bidirectional. You could also start with a large number of cells and add a treatment that causes death, and you could watch that change happening in real time. Another thing that you can measure with impedance are changes in cell morphology. So if you have a cell that is more compact, add a treatment that causes it to spread out, that type of thing can absolutely be detected by impedance with very high sensitivity. In fact, changes that you would have a hard time detecting by microscopy, you can actually see using impedance. And again, the process is reversible. If you add something that causes cell shrinkage, you can watch that process happening in real time. Something else you can measure with impedance are changes in the strength with which cells are interacting with the plate. And that may not be particularly intuitive, but if you take a cell that's lightly resting on the plate bottom, you compare that to a cell that has multiple focal adhesions and is being held very tightly to the plate bottom. So there's a high level of intimacy between the phospholipid bilayer and the plate bottom. That changes the ability of the electric current to flow between the different electrodes. Another thing you can measure are changes in barrier function. So this won't be true. I'm being told that maybe I'm not being loud enough. So let's see what we can do. Try and adjust my microphone a bit. Another thing that we can measure are changes in barrier function. So if you happen to be using a type of cell that forms tight junctions between neighbors, that has a big impact on the sort of the uniformity of this insulating layer of cells. And so as you go from cells that are not touching their neighbors to cells that are that have formed tight junctions, again, that has a big impact on the flow of current. And finally, something that we can also measure are cell invasion and migration. And so this won't be relevant to virology assays, but we can do scratch wound healing assays in the bottom of these plates where you generate an artificial wound and you watch cells move in to close that. So impedance's ability to look at a wide variety of cellular phenomena actually makes it a really useful tool for virology assays. And the reason for that is that when you look at a standard viral cytopathic effect, it includes 4 of the 5 things that I've just mentioned to you. Over the course of this viral infection, you typically observe changes in cell number, cell size, changes in the cell substrate attachment strength, and also changes in barrier function if you happen to be using a cell type where that's relevant. And so one of the things that we've seen consistently over time is that impedance picks up on viral cytopathic effect even before you're typically able to see this by microscopy. So if we look at a largely confluent monolayer of cells, there's a very large resistance in the flow of current here. And at early stages of the cytopathic effect, even before you've seen a change in cell number, just the loosening of the interaction with the plate bottom and the rounding of those cells will result in greater ability of current to flow through the system. And then, of course, as you progress through the viral cytopathic effect, you'll see cells beginning to lyse or detach altogether, and then current flows even more easily. So what I'd like to do now is talk about the specifics of the workflow and give you a representative example of a cytopathic effect, just to give you a taste for what impedance signals look like. So in this example, we have used A549 cells. We seeded 40,000 of these per well in our electronic plate. And this was then put into the xCELLigence instrument, and an impedance reading was taken once every 15 minutes. Over the first 24 hours, what we're looking at are really adhesion and proliferation of these target cells. We can then pause the experiment, remove the plate, and add, in this case, it was an oncolytic virus prepared at 5x serial dilutions, put the plate back in the instrument, continue monitoring once every 15 minutes. What I'm showing here on the y-axis is, again, the impedance parameter cell index as a function of time. And this first sample is the uninfected sample. So these A549 cells show 3 distinct phases in this impedance trace. First phase is really cellular adhesion, and this typically happens over the first 10 or so hours. Of course, the cells will touch the bottom within usually 30 minutes, but they spend the next 10 hours really attaching more strongly and spreading out, and that gives rise to this steeper phase of the curve. We then typically see a second phase, which is a bit slower, which correlates with proliferation. You can confirm all this by microscopy. You can see in this phase, typically the number of cells is increasing. And eventually what happens is that the cells reach confluence, and so the impedance signal will plateau. And the reason for this is that the entire surface area on that gold biosensor array has now been taken up. And so there's just no more gains to be had. So something that's really important to highlight is the fact that this type of detail, the kinetics and the magnitude of the signal, they really serve as built-in quality control metrics. The Nobel laureate Arthur Kornberg was famous for having stated in his 10 Commandments of Enzymology, he said, "Do not waste clean thinking on dirty enzymes." And the idea here is obviously that if you're using a dirty enzyme prep, it send you down all kinds of rabbit trails that lead to spurious conclusions. And we've seen over and over again, the exact same thing is true in a cell-based assay. If you're working with target cells that are unhealthy in any way at all, perhaps they're at too high of a passage number, perhaps the technician didn't split them regularly the week prior, and so they've been stressed and they haven't recovered yet, that absolutely has an impact on the quantitative information you extract from the assay. That's true for virology assays. And so what we and others have often done is use the untreated sample as a quality control measure to determine to make a go, no-go decision in even running the viral infection. And so that can look a bunch of different ways, but in this example, one of the things you might do is you might say that in order for us to even add virus at the 24-hour time point, there are two conditions which must be met. The first is that the cell index should be at a value of 4 plus or minus, for example, 0.1 or 0.2 at the 10-hour time point. And then the cell index must be at the 24-hour time point. It must have a value of 6, plus or minus 0.3, for example. And if it doesn't meet those criteria, then we're simply not going to run the assay. This ends up saving you quite a bit of time, especially when you consider that these assays often run for 4 or 5 days. It's better to find out early on if there's a problem than to have to let it run the full infection time course before you make that decision. So what I'm showing now is the addition of virus, again at 5x serial dilutions. In green, this is about a 1.2 billion fold dilution. There's really not much impact. In blue, 230 million dilution, not much impact. At around 47 million fold dilution, you're starting to see a killing phenotype. Again, we know that this drop in impedance actually correlates with killing because we can couple this with microscopy. And I'll talk about this towards the end of the presentation. As we progress through these viral dilutions, you're going to see a steeper, more rapid killing response. Something interesting begins happening around 75,000x dilution, shown here in aqua. And that's that these curve shapes begin taking on more structure. And that structure gets more pronounced as we go to higher and higher concentrations of the virus. By the time we get to 120-fold dilution, shown here in red, there's a very distinct biphasic killing curve. So the details of this oncolytic virus killing are not important. I'm simply showing this here as sort of a generic example of what impedance traces can look like. But I want to highlight that these traces are very much like a fingerprint. So they're specific to the virus you're using, and they're also specific to the target cells being used. And they can even change shape to some degree, depending on the tissue culture media that you're using and other parameters for the assay. So something to highlight is there's a very clear dose dependency in these killing curves. And I'd like to address now the issue of reproducibility. So you can run your data analysis directly on this raw data. Something that we commonly do is a process called normalization. And let me just highlight here that these 12 samples are virtually superimposable here over the almost 24 hours. And they should be, because they're identical. Prior to virus addition, all of these samples have 40,000 cells in a well. But you'll notice there are some slight differences here immediately prior to virus addition. And in order to really get the best sort of data analysis out of this data set, one of the things we would like to do is to normalize so that all of these samples have the exact same signal immediately prior to virus addition. This essentially lets us run a fair race, where after the virus has been added, it's the most accurate way of comparing between the different samples. And so you'll see here, as I transition from the raw data to the normalized data, we've given everything the same value at that time point, and we've arbitrarily set that value as 1. So I'll toggle back and forth between these. What I'm showing now is the standard deviation for an assay that was run in duplicate. You can see that the error bars in many of these, they're actually even difficult to see. The exception is that at very low concentrations of the virus, at later time points, you do begin to see larger standard deviations. And this is sort of a universal phenomenon we've seen. And I actually believe this makes good sense considering the implications of having very few virions per well. So this is an example of the type of reproducibility you get amongst technical replicates. What about biological replicates? Well, I can tell you for this particular oncolytic virus, we looked at, we ran, I think it was 12 to 14 different assays over the course of 3 or 4 months. And the EC50s we were getting, they varied just 2 to 3-fold. So both from a technical and biological replicate standpoint, the reproducibility tends to be very good. So what I'd like to do now, having shown you sort of a generic example of what these traces look like, is address the question of how we put these to practical use. And so we're going to look at 2 different examples relating to neutralizing antibodies. I think most of you are extremely familiar with this, but just in case, sort of the gold standard in this space would be to do a plaque reduction neutralization test. For this assay, you would have a virus of known concentration and an antibody sample, mix those, allow it to incubate for a given amount of time. And what you're hoping here is that the antibody has affinity for some epitope on the virion surface, and that binding event actually inactivates the virus, prevents it from having a productive infection. You analyze this by taking that virus and antibody mixture and adding it to cells. And typically your readout is going to be a plaque assay. So if you have very low neutralization ability, you're typically going to see the entire plate bottom getting blown out. If there's a little bit of neutralization activity, you might see dozens or a couple hundred plaques in your well bottom. And then very high neutralization activity, there's going to be a dramatic reduction, and you're seeing here, roughly 10 or 20. So of course, the level of neutralization you're getting is going to be dependent on 2 factors. The first is the antibody concentration. The other is the actual affinity of the antibody for the epitope of interest. So this entire process of the plaque assay can be replaced using xCELLigence. And so what that looks like is that you would have, here in this hypothetical situation, an uninfected sample. So the cells, again, are growing to confluence, and then they reach a plateau. You would have the negative control. So we're adding here the virus alone, and you're seeing a robust killing response. And then as you add in antibody, you're expecting a delay, shifting of that killing response. And the reason is because you're essentially adding fewer virions into this sample. As you add in more and more antibody, or the antibody has higher affinity, you're again going to see a progressive pushing to the right of this. And eventually the expectation, of course, is that you've neutralized all of the virus and you see no killing response at all. So let's look at an example from the literature where this assay was used to look at neutralizing antibody titers in wild specimens of West Nile virus. In this example, Vero cells were used, and you're going to see in a lot of the examples I show here today, Vero cells are used. So in this case, it was 50,000 of these per well. They were grown to confluence overnight. And then they were infected with West Nile virus at a multiplicity of infection of around 2. The West Nile virus, prior to being added to the cells, was pre-incubated with 2x serial dilutions of neutralizing antibody of known titer. This is the data. Again, the notion of a fingerprint. So these Vero cells behave very differently than the A549 cells I showed you a few slides back. Vero cells, as you probably know, are kidney cells from African green monkey. They have a fibroblast-like phenotype. And this is a very common type of impedance trace for fibroblasts, where they show a very rapid sort of increase in impedance and then a sort of a relaxation followed by another rise. But this, over the first 24 hours, is what the cells are doing by themselves. This is the uninfected sample. So you see that they've grown and they're still increasing a bit, but they're basically plateaued here. This is the point of virus addition. And this is the sample that was infected with virus alone that had not been pre-incubated with any antibody. So you see a rapid killing response that goes all the way down to an impedance value of 0. As you add in low amounts or intermediate amounts moving up towards high amounts of antibody, you see, again, a shifting of these curves to the point here where at the highest antibody concentration, it has almost completely neutralized the killing response. It doesn't quite look exactly like the uninfected cells, but it's very close to that. So how do we take this primary data now and then extract some sort of quantitative information from it? There's a bunch of different ways that you can do that. For example, one thing you could do would be to analyze the area under these curves as a function of the antibody concentration. In this particular case, what the authors chose to do is they've simply asked, how long does it take to get a 50% killing response? So they looked at the untreated sample and said it has a cell index value of roughly 1.4. So if we draw a line at half of that, at 0.7, at what time point does that line intersect with these different samples? They've then plotted that as a function of the antibody concentration. You can see here you get a nice linear fit. So the authors then used this standard curve that was produced with an antibody of known concentration to then analyze West Nile virus titers in wild birds. So again, what that looks like is to take Vero cells, grow them in this electronic plate, and then you're adding to this purified West Nile virus that has been pre-incubated, now not with antibody that's purified, but with bird serum. And so, for example, if you ran the assay and you found that it took about 80 hours in order to have a half killing response, then you could easily determine the antibody titer in that bird's serum. So what's very important is that when they use the impedance approach to determine antibody titers, they found very strong correlation between this and a traditional plaque reduction neutralization test. They analyzed dozens of different birds. I'm just showing 14 of those here, but this is the titer determined by xCELLigence, also known as real-time cell analysis. And this is the result determined by plaque reduction neutralization test. So you're seeing 40 versus 20, 1280 versus 348, 40 versus 15, 80 versus 20, etc. You can see that in many cases, the values are very close to one another. I'd like to now look at an example of trying to monitor the neutralizing antibody response that's produced by immunization. So in this example, 2 different subjects were vaccinated against influenza H1N1, also known as swine flu. And then at days 0, 7, and 21, the subject's serum was collected, and that serum was incubated with purified virus and then used to infect cells in one of the xCELLigence electronic plates. What I'm showing you here is the data directly from this paper. There's a lot going on, and it's also a bit blurry. And so I'm going to toggle back and forth just to show you that I went ahead and just sort of traced this to make this easier. I want to be clear that the traces faithfully replicate what's present in the primary data. So I'm going to now walk through this data, looking at the 2 different subjects. So this is the no virus control. In this case, these particular target cells are being grown long enough that there's a bit of a drop in the impedance signal over time. And this is what it looks like to take those target cells and infect them with swine flu. So you see a rapid healing response over the course of really about 10 hours. This drops down towards 0. So if you pre-incubate the virus with serum from subject 1, on the day they were immunized, you see that there's a very small shift to the right here. And obviously they haven't developed immunity yet to what they were vaccinated against. So there's sort of this just sort of generic neutralization associated with their serum to begin with. If you wait 7 days and then repeat the assay, see that this is shifted a bit more to the right, but it's not substantial. If you wait and analyze again at 21 days, you see that they've essentially completely neutralized the virus at this point. And this is consistent with what you would expect in terms of the overall kinetics of the response. What's very interesting is if you analyze run the same type of assay from patient number 2, subject number 2, again on day 0, there's a slight shifting relative to the virus alone control. But what's different is that for subject number 2, on day 7, they're already beginning to see neutralization. Now the virus is still able to end up killing, but there is this shifting, and this is indicative of the early stages of the immune response. In their serum, they have at least some neutralizing antibody that's effectively reducing the concentration of virus that's being introduced to these target cells, and that's why there's this delay. And then, of course, if you go out to 21 days, they have a complete neutralization response. So I'd like to point out that this xCELLigence assay is being used in a DARPA pandemic project. And so in this particular project, what's being done is they identify individuals who have survived and recovered from an infection by the virus of interest. They then isolate antibody expressing B cells and perform single cell sequencing of the antibody heavy and light chains. And they identify the correct sequences using bioinformatics. They then synthesize the heavy and light chain genes and express the protein and purify it. And this is where the xCELLigence comes in. They're able to, in a high throughput manner, screen these antibodies for protection against the virus of interest. And there are two of the higher throughput instruments for xCELLigence. There's either 576 or 1536 wells that are available for this type of screening. And then finally, of course, once you've identified the best candidates, the mRNA encoding for the antibody of choice can be delivered directly to the patient, and their body can make the antibody that's been selected. So I'd like to just kind of summarize this section of the talk. We've seen, in terms of antibody detection or quantification, you can use a standard curve. We've also looked at an example where the detection is just more qualitative. At the kinetics of an emerging immune response. It's possible to do very high throughput screening, and one of the reasons for that is that it's such a simplified workflow. So very minimal hands-on time. There's a plating of the cells. You're talking a couple of minutes here, assuming you're using a multi-channel pipette, and then there's an adding of the virus step. You don't need to generate plaques or count plaques. An important aspect of this is that there's an element of safety. If you're working with something that's particularly dangerous, the minimal hands-on time obviously reduces your exposure. In terms of data analysis, it's literally minutes. So there are a number of built-in software tools that would allow for you to analyze things like area under the curve, the cell index value at a given time point, slope, etc. And many of these tools have very nice sort of drag and drop features where, for example, if I was analyzing this data set here and I decided I wanted to look at area under the curve, it's simply a matter of dragging sort of a start and stop line to define what temporal window I want to do the area under the curve analysis within. And then the secondary data, the output of that analysis, is generated in seconds. So it's truly very, very easy to do these types of analyses. In terms of precision and reproducibility, You've seen a couple of different plots now where you get a sense for the size of the standard deviations in a technical replicate. Tend to be very small. And I also mentioned that the biological replicates tend to be tightly clustered as well. And with the West Nile virus, you saw that there's good correlation with traditional methods. And I would say that's universally true across the literature when you look at these different types of xCELLigence assays. So I'd like to move now to an example of using the impedance assay for virus titer determination. So if you think about typical virus titering, we buy a number of viruses here, and sometimes the titer is listed as the number of genomes that have been identified per mil, or maybe ELISA has been used to look at a given virion coat protein and its concentration, or maybe light scattering has been used to determine the number of particles. The problem with all these is that they're just not functional assays. I mean, that's obvious. So, for example, with light scattering, you may have a certain percentage of those virions, they may not have genome even packaged within them. Right? So this just really kind of highlights the benefit of having a functional assay that's easy to do. Historically, of course, for determining titer, the plaque assay has been the gold standard since, I think it's since been like 1952. With xCELLigence, the way that this works is that, at least in the variation I'm showing you here, one caveat is that you need to start off with a virus prep where you know the concentration of the stock. And with that, you can then prepare a standard curve that can be used for analyzing all subsequent virus preps. So you're again going to plate cells, grow them to confluence, and then infect these with serial dilutions of the virus. There is an alternative, and that's that you can do the infection in suspension and then plate the virus plus cell suspension into the E-plate and let the cell settle and then monitor the impedance response. And that's in fact what was done here. So in this case, again, there's 50,000 Vero cells per well. They're in suspension. They were mixed with 10x dilutions of West Nile virus and pre-incubated for 30 minutes. And then that mixture was added to the plate. xCELLigence was used for reading this. And so just to kind of orient us, again, here we're seeing the uninfected cells, so they're plateaued. And then this is adding the highest concentration of the virus. So this is a little bit unique, and it's a consequence of having done the infection in suspension. You'll see that this sample with the highest virus concentration, it begins to drop even prior to these cells reaching their maximum value. If you go from high virus to lower concentrations of virus, you'll see this progression moving left to right. So you're taking longer and longer to have a complete killing response when there's less virus around. Of course, that makes sense. So again, how you analyze this, there's a number of ways that work just fine, but here again, the authors chose to look at the time point at which a 50% killing response had been achieved. And so the uninfected cells have a cell index value of about 1.4, so half of that is 0.7. We're plotting the intersection time point here as a function of the virus titer. Again, you see that you get a nice linear response. And so, of course, you can then use this for all future preps of this virus. You just simply have to run a single killing assay, and that 50% response time will then tell you the titer of the virus that you've just produced. So something it's important to point out is that the slope of this curve is unique to each virus cell system that you're using. So for example, if I'm working with adenovirus, I would need to prepare a standard curve for wild type, and then for each of the different mutants that I'm working with, because each of those mutants are expected to have different killing kinetics. But what this saves you is having to do a plaque assay each time you've produced a new batch of virus. And so I would like to refer you to this paper, which is a really nice example of this virus titering done by a group at Sanofi Pasteur. So of course, these guys are in the business of vaccine development. And they made a couple of really striking statements in this paper. One of them is that when they compared to a traditional titer determination assay, the xCELLigence approach was 5 times less labor intensive in terms of operator time. It costs 3.5 times less when you consider operator time, the reagents, and the consumables. So on the basis of this, they said that the RTCA, or the xCELLigence assay presented here, is now being used for titration of viruses to support their bioprocess studies. So I think it's pretty striking when a set of researchers that are in this space have completely changed their workflow to adopt this. That's saying something. The next thing I'd like to move to is an example of antiviral drug discovery. So in this case, yet again, VeroCells. Trust me, xCELLigence works with pretty much any cell type you can choose. Just in the virology space, VeroCells are very much the favored model. So at the 50-hour time point, these cells were treated with either wild type or with a mutant of the herpes simplex virus 1. This particular mutant has a mutation in the DNA polymerase gene of the virus, and it makes it resistant to the drug acyclovir, which is a nucleoside analog. So 90 minutes after adding the virus, acyclovir was added at different concentrations. I'm showing here the data just for the wild type virus to help orient us. These are the uninfected cells. I wish these were in color, but I'm telling you that this larger dashes here is the uninfected sample. So note that in this particular assay, the conditions they've used, even without treating the cells with virus, there's a reduction in the impedance value. And the reason for that is simply that the cells are, they're getting sick on their own over time. And this could be due to depletion of the media or whatever it may be. But what's important here is that compared to the untreated cells, when you add virus, there is an acceleration of this drop in impedance. So this is virus in the absence of any drug. Now, acyclovir inhibits the herpes DNA polymerase. So as you add in drug, the expectation would be that you would mitigate this killing response. And that is, in fact, what's observed. As you go from low to high concentrations of this drug, you see these curves shifting upwards. At 2 micromolar of the drug, you've pushed the curve almost to the point of being equivalent to the uninfected sample. So they went to then try and determine the EC50 for acyclovir. And this could be done at any time point that you think is appropriate. They chose the 120-hour time point, which I think is quite reasonable because you have a robust response here, where the different drug concentrations are spread out amongst that range. When they did this, they were able to demonstrate that the EC50 for acyclovir for the wild type herpes virus is around 1 micromole. And importantly, when they repeated this assay with the herpes virus mutant, that again has a polymerase resistant to acyclovir, you can see that the EC50 increased around 100-fold, up to 100 micromole, which is exactly what you'd expect considering what's known about that mutant. So this gives you a sense for the way the assay can be used, not just to identify whether a drug has an impact, like in a high throughput screen, but to then pull quantitative information out of the assay. I'd like to now look very quickly at an example of viral fitness. So, when we talk about fitness, there's a lot of different ways you might define that. Like in some cases, people will look at, well, how many viral genomes are produced as a function of time or something like that. The problem with that is that it's not really looking at the entire viral life cycle, considering entry into the target cell, the production of viral genomes, the production of virions, the release of those virions, and continuing the life cycle over and over again. So in this assay here, they're looking at the ability of the entire viral life cycle, and from the point of infection to producing infectious virions that propagate the cycle. It was focused on viral hemorrhagic septicemia virus. This is a virus that causes death in fish, and it's particularly problematic in fish farms. And something that's interesting about this RNA virus is that at its terminus, they, by bioinformatics, identified that there was expectation of hairpin formation, and the authors suspected that this hairpin would have an important impact on the ability of the polymerase to produce copies of that viral genome. And so what they asked is when they made point mutations within this hairpin region, what the consequence of that would be on the total viral RNA that's present in infected cells. And so you can see here in wild type, in these 3 different mutants, this is a single point mutation, and then in green and blue, you have a combination of 2-point mutations. But red and blue are largely similar to the wild type, and then the green, there's a fairly large drop, maybe a 2x drop in the total viral RNA levels. So if you were to use this sort of biomarker readout as your assessment of viral fitness, you would draw one conclusion. But what's very interesting is when you look at the total killing process by this virus using the impedance readout, you draw actually a different conclusion. So shown in black is the killing response, again, dropping the impedance value towards 0 for wild type. Now, if you look at the red sample, this point mutation at position 8, you can see that its RNA levels are equivalent to that of the wild type, and yet it's actually a more efficient killer. Now, if you look at the green sample, which has the largest drop in RNA levels, it actually ends up being the least efficient killer. So you have this delay here of something like more than 50 hours relative to the wild type. What I find very interesting is that the blue sample, mutated at position 7 and 8, has a very similar level of RNA as the wild type, and yet its killing response is delayed by, I guess this is somewhere around maybe 25 or 30 hours. So this really highlights the value of doing a functional evaluation of virus fitness as compared to using a biomarker-based evaluation. What I'd like to do now as I begin starting to wrap up is to talk about combining impedance with live cell imaging. And so the goal behind this is really increasing the information richness of our assay. So at this point, there are a few thousand xCELLigence instruments that have been placed globally, and this has resulted in somewhere around 2,000 publications. This plot here shows the number of papers as a function of year, and fortunately, it's not quite up to date, but I can tell you that we're around 2,000 right now. And something that we've learned as we scan back through these papers is that our users are often using the xCELLigence impedance assay as the primary readout, and then coupling this with images for confirmation. And so the workflow for that is that you use what we call our view plate. So the very first xCELLigence plates had the gold biosensor array spanning the entire width of the well. And when we started realizing that people were wanting to do imaging directly in these plates, we developed many years ago this view format, allowing microscopy in the center of the well. And so in terms of workflow, you would use one of these view plates, and then at key time points, you would just pause the xCELLigence experiment, pull the plate out, take it to the microscope, collect your images, and then continue the impedance assay. And this works, but there's a lot more hands-on time, and it basically becomes an endpoint assay based on imaging. And so improving upon this was really the impetus behind our newest version of xCELLigence, what we've called eSight, and that's being shown here. The idea is that you would use a single simplified workflow, which is the same as what I've been showing you, but that you would get 2 data sets with unique perspectives. So you would have the real-time impedance coupled with live cell imaging. This is a view of the top of the instrument. You'll see 5 cradles here. All 5 of these are capable of doing imaging, and the 3 in the back do both imaging and impedance. The instrument can scan impedance in these different cradles independently of one another, so you don't need to try and coordinate with what your colleagues are doing at any given time. It takes about 15 seconds to scan the impedance for an entire plate, or a little bit less than 15 seconds. In terms of imaging, this instrument can use both 10x and 20x objectives. It can acquire bright field images and then also red, green, and blue fluorescence images. And it can generate movies. So what I'd like to do now is to walk you through the workflow for the eSight instrument. We're going to seed cells into the wells of the electronic microtiter plate and put the plate inside the eSight instrument, which is housed inside a tissue culture incubator. For most virology assays, you're going to monitor cell adhesion and proliferation over the first day, and then pause data acquisition, remove the plate from the instrument, and add virus to it, then put the plate back in and start data acquisition again. At this point, you're able to monitor two different types of output simultaneously. Using the microscopy viewing window in the center of the well, you're going to be able to capture live cell images. So this will include bright field as well as the red, green, and blue fluorescence channels. In this example image here, the red channel has not been included. At the same time, the gold biosensor array will continuously monitor impedance data in the same manner that I've already described for you. I'd now like to show you eSight in action using the example of the same oncolytic virus that I introduced at the beginning of the talk. So the A549 cells that were used as a target here express blue fluorescent protein that is localized to the nucleus. This helps for doing counting of the cells. If you don't have the ability to engineer your cells to express a fluorescent protein, we sell a variety of dyes that are compatible with live cell imaging. The oncolytic virus that's used here expresses GFP. And it's important to note that this virus, rather than lysing the target cells, causes them to fuse to one another. This process occurs serially to produce very large polykaryocytes that eventually cease to metabolize. In this first example, we're going to look at the uninfected sample, this purple trace here. So at 24 hours, the cells look quite healthy. They're very near to being confluent. You can see a couple of regions of daylight between these cells. If you move out to the 72-hour time point, you can see that the cells have continued to proliferate and they've just packed in tighter and tighter next to one another. By 112 hours, the cells are starting to look pretty unhealthy. They're beginning to stack up in 3 dimensions. I'd like to now contrast this with the case where the cells were infected with the highest concentration of virus at an MOI of about 5, which again is this red data trace shown here. At 24 hours, the cells are again quite healthy and confluent, but just 6 hours after adding the virus, you can see a very robust cytopathic effect. So big regions of daylight have opened up between the cells, and you can see cells beginning to fuse to one another. When you move out to the 62-hour time point, this cytopathic effect is even more extreme. You see very large fusion bodies, and you're starting here to see the green fluorescent protein that's encoded by the virus. So it's very clear that there's a strong correlation between the impedance signal and what is physically taking place with the cells. It's quite striking to watch the viral infection process dynamically. So I'd like to now show you this in movie format. Again, using the sample that was infected with the highest concentration of virus. It's worth pointing out here that we began collecting images immediately after the cells were seeded in the plate. And so because of this, over the first few frames, you'll see that the cells are quite rounded as they haven't attached and spread out on the plate bottom yet. Seeing attachment, proliferation, and right here is when virus was added. Starting to see fusion, getting to see the GFP encoded by the virus be expressed. At late time points, the cells are basically just collapsing into piles of debris. So we know that they're not attached to the plate bottom anymore because the impedance signal has dropped all the way down to 0. So despite the fact that there's so much material in the well bottom, there's just no longer intimate contact with the plate bottom. I find it instructive to watch this same infection, but to do so with only the blue and green fluorescent channels and to remove the bright field channel. The virus has now been added, beginning to see cell fusion, starting to see the GFP from the virus show up. And eventually everything just shuts down, no more activity. I'd now like to draw some conclusions, and this will include key points from the entire talk, but also I'll give some summary points relating to the combination of impedance with imaging in the eSight instrument. So we've looked at 4 different applications, including neutralizing antibodies, determination of viral titers, antiviral drug discovery, and evaluating the relative fitness of different virus strains. So very clearly there's a broad applicability of the impedance-based assay and also very high sensitivity. And these attributes are related to the fact that we are simultaneously monitoring changes in cell number, cell morphology, cell substrate attachment strength, and when relevant, barrier function. It's worth pointing out once again that impedance is capable of tracking changes that are caused by the virus even before those changes are detectable by microscopy. We've seen that over and over again, not just in virology assays, but in diverse impedance assays. Reproducibility of the impedance assay greatly exceeds what's possible using traditional methods, whether those be plaques or colorimetric assays that look at cell metabolism, for example. So in the case of the oncolytic virus that I've shown, we did, I think it was 14 replicates, biological replicates, over the course of 3 or 4 months. And we found that the EC50 we were able to calculate using impedance varied just 2 to 3-fold. In contrast, the legacy assay that had been used for that virus was an Alamar Blue assay. And there, the biological replicates varied 100-fold. So impedance was able to provide a very substantial improvement in the reproducibility of that assay. The impedance assay has a very simplified workflow. The hands-on time is really just limited to adding cells to the plate and then adding the virus. Any sort of subsequent processing of endpoints is entirely eliminated. And one of the benefits of this is, of course, that it reduces your exposure risk. In terms of throughput, we offer a variety of xCELLigence instruments that vary anywhere between 16 wells all the way up to 1536 wells. So continuous monitoring using impedance reveals subtleties that are missed by endpoint assays. And the oncolytic virus assay is a good example of this. This type of variation in the killing response as a function of virus concentration, this is not something that you would pick up on very easily doing an endpoint assay. So imagine comparing this continuous data trace to an assay where maybe you took endpoints at 24, 48, and 72 hours. You just really would not have any sense for the subtlety of what's going on here with the endpoint assay. Finally, when you combine the impedance with imaging, one of the benefits is that this really maximizes both the sensitivity and the specificity. So with the ability of impedance to look at changes in cell number and size and attachment strength and barrier function, as we've already said many times, there's very high sensitivity associated with that. The challenge is that because it's a composite readout, you don't always necessarily know what it is you're looking at. And that's where imaging gives you specificity. Those images make it clear whether you're changing cell number or the cells are swelling or shrinking over time. So you really get a more information-rich assay by combining those two. And finally, with the eSight instrument, it's nice to have a primary and a confirmatory output all from a single assay. The gold standard in science is obviously to get a result and then try and confirm it using an orthogonal approach. With eSight, you get to do both of those in the exact same well. It's the same population of cells that are being interrogated both by impedance and by imaging. And lastly, what I'll say is I haven't really said too much about the reagents, but we sell a variety of reagents for the eSite that facilitate live cell imaging. And these include dyes that stain the nuclei of cells, the lentiviruses for expressing fluorescent proteins, and then also reagents designed to detect cytotoxicity or apoptosis. And that list of reagents is expanding continually. With this, I'd like to very quickly walk you through some resources. We have a virology research grant that's open at the moment. The purpose of this grant is to help scientists achieve their research goals by providing them with access to our xCELLigence technology. So for 6 months, the grant winner gets access to an xCELLigence instrument, free consumables, and then also our consultation to help you get up and running for that 6-month period. The deadline for this virology grant is about a month from now, and the winner is announced 2 weeks after the deadline. So I encourage you to submit an application for this. If you visit the website, you can see past winners of this grant. So this is where you can get information about it, and including instructions on how to download the application and submit it. Finally, we have a virology handbook that includes many of the assays that I spoke about today, and I would encourage you to take a look at that for more information. The xCELLigence instruments are shown here, including the newest eSite instrument on the far right. If you'd like information about each of those specific instruments, you can find that on our website. So we are now part of Agilent, but our website has not been fully merged yet. And so at aceabio.com, if you go to the products tab and hover over that, you'll see the full list of xCELLigence instruments. It's worth noting that we also sell flow cytometers that are quite innovative. And so I'd encourage you to take a look at that as well. The breadth of what's capable, what xCELLigence is capable of in the virology space is really just a small window into what the instrument is capable of doing. It's being used throughout the world in dozens of different applications that span everything from cancer and immunotherapy to stem cell work to even toxicity testing. And if you'd like to get a sense for some of the other things that xCELLigence can be used for, I would encourage you to, again, go to aceabio.com and look in the applications tab. You'll see some of those listed here. With that, we're going to go ahead and open it up for questions.
Thank you very much, Brandon, for that excellent presentation. We've already reached the top of the hour, but we'll try to get at least a few questions in. And if we don't get to your questions, we will make sure that we forward this to Brandon and we will email you the answers to those questions. So let's go ahead and get started. So Brandon, you showed data from both impedance and imaging. Do these two approaches usually give similar quantitative data?
Yeah, that's a great question. So I'd actually put together just a quick table to address that. So I'm showing here, I think this is 5 different types of assays. So oncolytic virus, apoptosis, some CAR-T mediated killing, cell migration. This is an assay looking at activation of something called the unfolded protein response. And then I've shown the EC50 values here using either impedance or imaging. What you'll notice is that these two approaches often give very similar values. These values here tend to be, twofold or less different from one another. In the case of the oncolytic virus, it was between, roughly 3 and 7. So I think that this is actually a quite good correlation when you consider that these are cell-based assays. And I attribute this similarity to the fact that you're analyzing cells that are in the same well. It's not a different plate and a different instrument and a different incubator on a different day. It's the exact same population of cells.
Great. Thank you, Brandon. The other question that we have here is, you've shown that in a number of the examples where the virus is being added to confluence cells, the question is, why do we have to add the virus at confluence? Can it be added in the growth phase?
Yeah, absolutely. So many of the examples I showed today are from literature, and I can't speak to what the author's rationale was, but you can add virus at any point, you know, as long as there's enough cells in the well bottom to actually be detectable by impedance. And I would say that for most cell types, you probably need, you know, at least 5,000 or 10,000 cells. You're gonna be able to see a response. And so being confluent is not a requirement for the assay to work.
Okay, and can you talk about the sensitivity of the infection assay by xCELLigence system?
So it's a little bit hard to address, you know, what exactly is meant by sensitivity, but let's address it in terms of how low of a multiplicity of infection can you go to and still see a response? So the answer to that question is also dependent on the duration of your assay window, right? So in the case of the oncolytic virus I showed, we could dilute that virus out like to, you know, it was around 1 billion-fold dilution. And you're talking there about having a minuscule number of virions per well. You can still see a killing response, but you just have to let the assay run a bit longer, you know, for the virus to infect a cell, and then for those progeny virions to then propagate across the well bottom. So it's hard for me to put a number on sensitivity, but I would say, yeah, it's actually very, very sensitive.
We'll go ahead and take one last question here, Brandon, just because we're 5 minutes past the hour. And like I said, the questions that we didn't get to, we will make sure that we have a response and we will email them to you. The final question is, Brandon, what causes the spikes in impedance that's commonly seen at the time of when the experiment is paused or when the virus is added to the plate? Is it a function of media, temperature? If you can comment on that, that would be good.
Yeah, that's a great question. So when you make a change to the well, whether that's adding a drug that's in DMSO or adding virus, there are obviously two things that are happening. One is you're changing the composition of the medium, but then you're also having some sort of a change in the cell's health and behavior. So if you talk about just the change in the composition of the medium, something like increasing the ionic strength, for example, you could see a change in impedance. But what's important is that you just use the appropriate control. So if you're using a drug that's in DMSO, you add the DMSO vehicle by itself, you're going to see a small change. It's important that you compare everything else to that control. In terms of what's happening with the cells, we often see that the cells, whether it's a drug or a virus, will begin to swell as kind of like a stress phenotype in the very early stages. And that gives rise to a spike in impedance, which is usually transient. And so the cells know something is wrong and they're trying to respond to it. And prior to going into a full-blown sort of death cascade, what you're seeing there in the first few hours is usually cell swelling.
Great. Thank you, Brandon. We have a number of other excellent questions, but as I mentioned, unfortunately, we will not be able to get to them today, but we will definitely formulate an answer and then send those to you via email. So thank you again, Brandon, for your presentation, and thank you all to our excellent audience today. And please join us in future webinars. We'll have a number of additional webinars focused on virology and virus applications. Have a great day.